Hello, Professor. My name is Nidia Apostle. This is the video for the class Bio 225 Microbiology Week 3. So the first thing that I want to say is we are in the time we're going to be using our microscope. So the type of microscope that we're using is the HOL 600 and is a compound microscope. And we are going to go over the functions, the parts, and this is basically the video for the lab, and it's titled Use of the Microscope for Microbiology class. So here it is. I'm gonna turn my camera so I can show you the pieces and how to use the microscope. So here is my microscope. So the first part I want to show you is that the microscope needs to be handled appropriate. So this part here is called the arm, where I'm holding the microscope. That's the arm, and the other hand should be in the base. The base will be the bottom part. So one of the hands should hold the base, which is this, and the other hand should be in the arm, so you are able to hold that appropriately. So let me just get a little further so you guys can appreciate what I'm saying. There, I think that's much better. So hold it through the arm, the other hand goes under the base, and you lift and hold to manipulate the microscope in the right way to not break it or hurt it. So the first part is the ocular or lens or eyepiece. So the ocular or eyepiece can be different strains. The one that we're using is the 15 times. So this is your piece that is called ocular eyepiece. And then we're gonna move to the tube. The tube connects the ocular view of this piece all the way to the bottom here, where you're going to encounter the objective lenses. Now, the objective lenses are different strains, and usually it shows the numbers and the strains in each of the lenses. Okay, so the terter, this part here, the round piece that holds the eyeglasses, allows you to move them and manipulate them so you can have different views. But just to give you an example, so let's go here. So this is the 4 times lens. So you're gonna take this 40 times lens, multiply by the 15 to get your total view or magnification of the specimen or object that you are looking at. So in that way, you know what is the total. So that is the 40 and the 15. So that will give you the amount that you are looking at. If this piece will be 10, then you will say 10 times to the 40, it will take you to the 400. But because it's 15, it will be the 600. Now this is the 600 microscope, remember that. Now we have the oil immersion that we work with, and that will give you 100 more of the magnification to view an object. So we'll talk about more as we do our experiments. So let's move to the stage. This is your stage. This is the clips that holds your specimen. The clips are very important so they can hold your specimen when you are looking at the object. Now, how do you will move your object in the event that will be too close, too far, too back, or too forward? So I'm gonna move to the side. To the side, you can appreciate 2 knobs, the one in the back and the one in the front. So the one in the back makes the clips to go forward, so they come close to you, or they go backwards. Okay, now if you need to move to the left or to the right, either way, you use the knob in the front. That's gonna move your specimen side to side. Okay, now if you appreciate, in the middle you have your lens that is going to allow you to view your specimen. So you can see it right there. That lens, you need to clean it very careful with one of our little papers that they give us. Do not scratch, just softly do it. But that's what allows you to view your object. Now we have in the bottom the illuminator. The illuminator it's a little box, and you put your battery in the bottom, then you insert the light in the middle, and now it takes the light to see your object. So that is your illuminator. Like I said, this is your base. Clips of the stage. Now, a part that I love is the course. Basically the course adjustment allows you to clear your view, to put it closer, to put it further. And the fine adjustment does a fine focusing. So course adjustment, move the stage up and down and to focus better your view. And the fine stage, it's a fine focus that can intensify and make more intense the picture that you're looking at. Okay, so that's the pieces of our microscope, and now allow me to show you one of the samples that we're going to be viewing. So here it is. You can see there is a piece in the microscope. This is our boxes that are labeled with the different objectives, bacteria that we're going to see, like the amoeba, whole mount, and so forth. So we have a couple of them that we view through the microscope. One thing that I want to clarify is, and this was a hard part to learn actually, the depth of the image. So what is the difference between the depth and the field of view? So the field of view, you can see it's right there in the middle. That little circle, that is your field of view. And what the field of view is the part of the object that is being examined. So what is the space for that object that we're looking at? And you can measure it with your ruler like we did to calculate how many mm's are. So if you look at it under the 40, it's approximately 2 mm's that you have to, that's basically your object space. Now, when we talk about the depth of focus is, see this part here? You're looking at your object through this. How deep you have your object. It's a basically image space. How close and how far you can adjust the image. So when you go with the coarse, you can put it closer, you can put it further. The idea is of the depth is amount too close, amount too far. So that's the depth of the image. So there's two different things, the space of your object and the depth. How close, how far I am from that image that I am examining. So image space, object space. So that's how is basically the difference. So I did a couple of pictures. Let me show it to you. This one is the E at the 40. And then let me show you the next one. And that is the E to the medium power. And you can see, you can hardly see when you're in the high power or even with the oil immersion. You see all the oil, but you don't see really nothing there. So that is the E. And here it is. This is my amoeba slide that I view through the microscope. Maybe, depending how you move the picture, you have to look for the best slide that you can get. Then this one is the penicillium. Penicillium slide. It was amazing to see it. I enjoyed really so much this little one. That was amazing to me. Then move to the next one. The next one was the gist. The gist, it's just like a little bubbling. And then I have the spirillum. The spirillum is like if you have a bunch of little things, like a little larvae or lawn. It was amazing to me to look at the microscope. It took me the whole night to observe it. Bacillus slide. This is amazing. It's like a little bubbles of water, or I don't know, like if you have a blister or so. And then picture 10, of course, that's the coccus slide. The coccus slide is also amazing to me. I thought it was so surreal to see these bacteria. So there it is. That's the video of how to use your microscope. Again, this is the microscope 600X compound. And until the next video.